Hello, I'm Dr. Janet Sumner. I'm a scientist, but I'm not a biologist. So I'm joining Dr. Rob Saunders in the School of Life, Health and Chemical Sciences for a tour of one of the labs. In this short film, we're bringing you a unique 360-degree immersive experience. So take a minute now to have a look around and see what you can see. Right, Rob, where are we going to start?
Well, we've chosen this lab because it's in many ways the hub for what we do in the department, this kind of laboratory. And this is a laboratory which is open plan, as you can see, and ordinarily houses members of several research groups doing basic biochemical and molecular genetic research. So much of the material that we work on here comes in from elsewhere in the department, from other laboratories, for example cell culture laboratories, which you can find out a bit more elsewhere on the website. The material comes in and really I suppose it's fair to say that a lot of what we do involves the analysis of specific biomolecules. So the first thing that happens is you might want to be fractionating your sample to separate different cellular components. And for that, you would typically be using facilities such as this, which is a set of different centrifuges.
Right.
So the centrifuges separate biological particles and materials on the basis of fragment size and density.
Is that why you've got different sizes of centrifuges
Yes,
then?
that's right. So some of these things will operate at different speeds. So the higher speed will be for separating smaller, less dense fragments. The individual rotors, you can see on the bench there, a purple rotor, which is moderately sized volumes of samples, and the big black rotor at the back would hold large tubes that you might be separating, pelleting cells from a large cell culture experiment.
Why would you need a kind of big bucket of stuff?
Well, depends what it is you're looking for. If what you're looking for is quite rare in the sample, you've got to grow up a lot of cells to get it. If you're doing something like separating DNA from bacterial cells, typically you're growing those in half-litre cultures, so it's quite large volumes.
Yeah. Okay.
So as I said earlier, the laboratory is used by members of several different research groups, but all of their activities really use very similar basic laboratory equipment. And this is a typical bay from one of these for one of these research labs, with lots of little pieces of kit dotted about. And you know, this stuff ranges from the whirly mixers for mixing up your samples. It makes a rather good
nice sound
whirring noise. Yes. To smaller centrifuges. So this is really a very small scale version of what we saw in that room there. And this would be used for separating very small samples in small 1.5 milliliter plastic tubes.
Right, that's the things that you see the people pipetting things into
That's
all
right,
the time.
so that's tubes such as these, and you'd be adding small volumes of liquid from small handheld pipettes there.
Okay.
And down at the end we've got a variety of heating blocks, so a lot of what we do involves incubation of samples at different temperatures, often because there's an enzyme involved. So many of the enzymes we use operate at 37 degrees, which is standard body temperature. But sometimes not, sometimes lower temperatures. And of particular note we have a large number of these machines, which are polymerase chain reaction machines. So polymerase chain reaction, or PCR. Is a very important technique in modern biology. It enables you to amplify very small amounts of DNA into the quantities of DNA that you're able to analyse by the kind of techniques I'll show you in the next bay.
I figured it had got something to do with DNA.
Yes, well, it has got a bit of a big clue on here. But this is a really important technique. It's the sort of thing we use in the fly lab for identifying which mutations are present in our flies. Right. You would use it for seeing if particular oncogenes are mutant in a tumour sample.
So we're talking cancer.
We're talking cancer research. And you know, in external laboratories, it's a very important technique in forensic biology. If you're trying to track down the origin of a hair sample at a crime scene, for example.
Right.
That's how you do your DNA fingerprinting.
Should have worn my hairnet.
Yes. So I've mentioned that what we really look at here are a variety of biological molecules, and we separate biological molecules by gel electrophoresis in many cases, and that's either using agarose gels for DNA, and you'll see one of those in just a minute. But also we separate proteins on a different kind of gel matrix. And essentially what you do here is you apply your mixture of biological molecules at one end of a gel matrix and pass an electric current across. And you separate the molecules on the basis of their size and possibly their charge, depending on how you're doing the experiment. So obviously you've got a mixture of biological material in that sample. And
so that's stuff you're interested in and stuff you're not interested in.
Yes, exactly it.
Right.
And so one of the ways that we can do this is to use radioactive tracers. You can specifically label molecules with low-level radioactivity. And we have an area in this lab which is closed off, obviously, because you need to have it contained away from general laboratory activity. But typically you can spot your biological material by exposing your gel to a piece of X-ray film, and the radioactivity causes the X-ray film to darken. That's the it in brief. Of course, more recently and more powerfully, I think, you can recognize specific protein molecules by using antibodies, which are proteins derived from the mammalian immune system. So these things recognize specific regions of characteristic regions of a protein, so you can unambiguously identify a protein within the mix.
And safer as well.
Yes, safer as well, because you're not dealing with radioactivity. There's less hassle in the bureaucracy as well, I have to say. Here we've got one such an example of a kit for gel electrophoresis. This is a medium-sized agarose gel, so it's horizontal. You can just see the gel matrix as this milky-coloured slab.
Yeah, it's like an agar plate.
Yes, it's really chemically it's the same, but it's a higher degree of purity. Oftentimes you have to extract DNA from a gel like this for further analysis, so it needs to be quite pure matrix. And it's in a tank which is filled with a conductive liquid buffer, which allows the electric current to be transmitted through the gel. So you'd typically be using that to look at the outcome of a polymerase chain reaction.
Right, so it's come from over
there
to here.
And you'd be looking at it on here. And agarose gels come in a whole variety of sizes. That's quite a large one, and this is the gel plate, which is quite a bit smaller, as you can see. So this would be the sort of thing you would use in a very quick analysis if you wanted to see if anything had come out in your PCR reaction. So
what kind of things are you looking for on the gel plates then?
Well, typically, for example, from the PCR, you'd be looking to see, well, A, did the polymerase chain reaction actually work? Have you got anything to see? And secondly, have you got the DNA fragment that you would be expecting to see? Is it there? Is it there in the right quantities? Is it the right size? That's the sort of thing you're looking at. You visualize the DNA by staining with a fluorescent dye, which shows up, glows orange under ultraviolet. So you need the right amount of DNA
to get the signal,
to be able to see if you've got anything there. There's we've all done experiments where you've not had enough sample to see what it is you're trying to see, and you can use a device like this which measures the absorbance of ultraviolet light in your biological sample, so you can see, has your solution got enough DNA? Have you got enough DNA there?
Is it worth carrying on?
Is it worth carrying on? Yeah. Or do you go back to square one? Similarly, you can use it for assessing the amount of protein present. And then finally, down in this bay, we have again, it's a number of standard laboratory pieces of kit. So we have the usual fridges, freezers, and very low temperature freezers here. So this one's more like a domestic freezer. It goes down to -20. That's kind of what you'd be storing your pork chops in at home.
Right. Okay.
This thing's -80 degrees, which is quite a lot colder, and a lot of you would use this for long-term storage of materials. For storing living cells frozen, you'd be using liquid nitrogen, which we don't keep in the main lab because it's continually evaporating. Of course, we have a separate lab.
So when you say long-term storage, what could you keep in there and for how long?
You could keep frozen bacterial samples alive in there indefinitely. You could keep DNA samples in there indefinitely. DNA is a very stable molecule, that's why it's useful for forensics. Actually, if you've got it in the right pH, the right buffer, it'll last for a long, long time. But this bay here also it has the sort of stuff you would have for your initial analyses. We've got stereo microscopes for doing dissections of starting material, if you're working with animal tissues, for example. and the gel kits, the usual thing. So that's a brief a whistle-stop tour of a molecular biology laboratory. It's in many ways, as I say, it's the hub of what we do in the laboratory. We've got a lot of other specific facilities which you can find out a lot more about by looking elsewhere on the school website.
Rob, it's been fascinating. Thanks very much for giving us the tour.
Well, I hope you find it interesting.